Okay, so like I said in the last video, ecologists haven't been that good at predicting the structure of communities, right? We just don't understand, beyond invoking some random process, why some dunes are dominated by jack pine and others are dominated by oak forest. We just don't understand what causes those two things. So instead, what ecologists often do is try to reduce that complexity in some way, right? So if you have a community that has 100 species, well, that's 100 dimensions you have to understand. But what if we could just say, oh, this community has 100 species and this community has 50 species, and there may be something different about that number that we can understand, right? So that's the concept of species richness. Right, so if we take a look at this picture, right, and we look at all of the sort of purple and yellow flowers, right, we could say there's two, species richness is 2 if we simplified it that way, right? But we could count all the different species in here, and in fact that's what you're going to do in lab this week, and that would be species richness. It's just the number of species you find in a defined area. That's the simplest way of describing a community. We just say, hey, how many species are there? Cool. But you're destroying 3 kinds of information with species richness. One of those is abundance, right? So the reason why I've got this purple thing up here is because there's way more of the purple asters, purple coneflowers, than there are of whatever the yellow species is, probably some kind of lactuca. We're also missing composition, right? A different field might have the same number of species, but a completely different set of species, right? And so you might have a completely different field. It has the same number of species. Maybe it even has the same relative abundances of all the different species, but they're all different species. So we've lost that information about composition. And we've lost a lot of the information about how those organisms are interacting with other. So for example, feeding and cooperation, those kinds of things we could put in that matrix I showed you. So let's look at these different ways. Abundance, composition, and interactions, different ways of studying those. Right, so how do we study abundance? Well, there are a couple graphical ways to do it. Species abundance communities tends to be log normally distributed. In plain English, what this means is that a few species are very abundant, but most species are rare. And you can get something called a rank abundance curve that I think is actually a useful way of visualizing a community. So all you do is you take the abundance of the species. Here it seems to just be the log count. So this species here has, close to 100,000 individuals. The species at the end only has 10 or so. I guess this is a log scale. And then you just rank them to make the graph not look like chaos. So literally this is the least abundant species and this is the most abundant species. And when you do that, you get the structure, this rank abundance curve. And what this does is it shows you information about abundance. And it gives us information about something called the evenness of a community. This describes how close in abundance all the species in a community are to one another, right? So if we look at these 2 forests, represented by 2 lines from this paper in the literature, what we see is that, the yellow forest has fewer species, the richness is lower. The brown one has much higher richness, but there's also, the brown one has higher evenness. Even though there are more species, that most of them are more similar to each other in abundance than they are here, where they're wildly different in abundance. So richness is the simplest index of diversity. There are other indices that incorporate abundance in different ways. So for example, the Shannon-Wiener index looks like this. You do not have to remember this. I probably won't even ask you to work with it. I just wanted to show you what it looks like. So the idea is that PI is the abundance of species I, and then S is the species richness. So you literally take the abundance times the log abundance and sum this up across every species in the community and make it negative, and that's the Shannon-Wiener index. So what this does is it gives you a single number like richness, but those numbers now will be different depending on the abundance of the communities. And there's quite a few of these that you might learn if you take a third-year plant ecology class that we have. And they all have pros and cons, which I think aren't relevant in this freshman class. But just know that they exist, that these indices of diversity can exist. And they often use information theory. So this is actually from information theory. It has to do with entropy and an information system. Okay, so going back to these 2 forests in Virginia, we know that they differ in richness and they have different patterns of abundance. But what about the composition? Right, so maybe are these first 10 species that the yellow community only has 10? Are these the same species in these 2 forests? Or are they different? Or, are these 2 species the same species or are they different? Right? There's information there that would be useful to know. do these forests have any species in common at all, or are they completely different? Are the first 10 the same, but the last 10 are not the same? We don't know that from this picture. So remember we talked about how a community with S species can be considered to be S-dimensional, right? That's, so for the Ross Reserve, just considering the trees, that's 50-dimensional. If we bring in all the other organisms, we get hundreds and hundreds of dimensions. And so if you remember, we used trait ordination, this principal components analysis, to take 6 different dimensions and reduce them to just 2, that we could interpret the relationships among the different traits. Well, you can do the same thing with communities to compare those that differ in composition, richness, and abundance. And I just wanted to make sure, I should have done this before, I've shown you enough old dead white dudes that I thought I would show you probably one of the world's best ecologists, Sandra Diaz. This is what she looks like. This is her work over here. She's actually an Argentinian scientist. I think Nature named her as one of the top 5 scientists to be watching in the world. She's just a really awesome, brilliant ecologist. And she's still alive, unlike all those old white dudes. So here is a different kind of ordination. This is called non-metric multidimensional scaling, but it works just like principal components analysis. It shows you communities of 50 species, but now reduced into 2 dimensions, just to show you those correlations again. And it shows you 3 different types of communities that this was sampled in. So what you can see is you have 2 mixed grass communities shown in blue, and this fescue-dominated species marked in green over here. And what this is showing you, just like the traits, is that these communities are sort of opposites. They're negatively interrelated with each other, and they're mainly separated on this axis here. And so they have some species in common, right? So species over here are ones that are only found in the fescue community. And species over here are ones that are mainly found in the mixed grass community. And species in the middle are ones that are shared between them, right? Remember that whole idea of Clements and Gleason, right? We shouldn't have any species in the middle if Clements was right. But we see quite a few, like Thermopsis rhombifolia. It's a type of legume that occurs in both communities. But Poa pratensis, which is Kentucky bluegrass, probably an invasive species. It seems to only grow in the fescue-dominated systems. This is probably what your front lawn is made out of. Right, so the location, which are the sites in colors, tell us which communities are similar. So each one of these squares is a different place where it was measured, and these ones are all very similar to each other. And these different sites that were measured are all similar to each other. And the location of species, marked as plus signs, tell us which communities they occur in. And that's how you read this. And I'm probably not going to ask you to read this the way I might with a trait ordination, but just know that there are ways of reducing that dimensionality beyond species richness or an index of diversity. You can use ordination, and that brings in abundance. It brings in composition as well. So we can tell that these species or these communities differ in composition. The last thing that none of those methods include so far are the interactions, right? And we can use network theory, which again, I'm not really going to show you in this class. But ecological interactions form networks that have certain mathematical properties, right? A network can let us describe richness, abundance, composition, as well as the interactions. So here's an example of completely made up interaction network. So this is completely fake. But what I've done here is I've shown you a bunch of maybe plant species and a bunch of different pollinators, again, completely made up. You can see which ones interact with which other ones. We can see the composition in terms of their names. And what I've done is I've used the size of the circle. So this is a very common species, and this is a very rare species. So these networks probably have the most information about a species. We have richness, we have composition, we have the abundance shown potentially, and we have all of the potential interactions. Now, of course, a real community that's many hundreds of dimensions that can get really, really, really crazy. So this is an actual network that someone described between many invertebrates and birds with plants in the middle. So this has a lot of information that is incredibly useful, but network theory is even more complicated than ordination, and I'm really not going to expect you to understand that. But just know that you can use network theory. Another problem with communities, which we're going to address in the lab this week, is the fact that they're scale dependent, right? So if we imagine we went and we dropped a quadrat, like we're going to do in lab, that was this big out here. Well, we would only find 5 or 6 species inside of this area. Versus if we dropped a quadrat that was this big, we might find closer to 100 species in this picture, right? So the problem then is that any of those methods of describing communities change if you measure different sizes of area. the most obvious thing is that as you increase the scale of the area you're looking at, you find more and more species. And there really isn't an easy answer to this problem. This is probably one of the main things that is a problem in community ecology that's not discussed enough, in my opinion, right? So what area should we sample? What happens is actually the number of species you find increases logarithmically as you add more and more area. So you find this is actually, some people talk about this saturating, but it doesn't, right? It's log linear. So this ends at infinity. So the problem then is that there really isn't a solution. And so the areas that people sample tend to be areas that are convenient to the size of our bodies, right? So it might be easier to bend over and look at a square this big than it would be to walk around all day in a square this big. And unfortunately, that's how we study communities right now. And I think that really limits our understanding. So we'll stop the lecture there, and we'll talk about talk about communities some more on the other side.